Hello everyone, this is Manogna and in this video we are going to talk about enzymes used for cloning in the field of genetic engineering. So what are cloning enzymes? Cloning enzymes are a group of enzymes used to facilitate the cloning and manipulation of DNA. These enzymes play a crucial role in various steps of DNA cloning, including DNA amplification, DNA fragmentation, ligation, or DNA modification. Here are the 8 enzymes listed which are prominently used for cloning. First up, we have nuclease enzyme. Nucleases are enzymes which are used to cleave phosphodiester bonds in DNA molecules. We all know that DNA molecules contain both hydrogen bonds and phosphodiester bonds between the nucleotides. So these nuclease cut the phosphodiester bonds between the nucleotides. And the function of nuclease is non-specific. We have two types of nucleases here. One's exonuclease and others endonuclease. So exonucleases are enzymes that degrade DNA molecules by removing nucleotides from either ends. We have examples of exonuclease such as lambda exo, exo3 and bal31. Let's get into the details. So here we have a lambda exonuclease which cuts at the 3' end and the exo3 exonuclease cuts at the 5' end and the bal31 exonuclease does the function of both the lambda exo and exo3. It cuts both towards the 5' end and the 3' end, leaving us this part. Coming to endonucleases, as the name suggests, endonucleases cleave DNA molecules at internal positions rather than the ends like the exonucleases. To cut the single stranded RNA, we use S1 nuclease and MUC1 nuclease. So the S1 cuts at the 3' end, while the MUC1 nuclease cuts at the 5' end. To get the double-stranded DNA, we use DNAs which are specific and restriction endonucleases which are non-specific. So next up we have polymerase. Polymerases form phosphodiester bonds in DNA molecules so that they can extend the chain of DNA. We have many polymerases such as DNA polymerase, RNA polymerase, etc. So DNA polymerase synthesize the DNA. The DNA polymerases are also employed in several applications such as PCR and DNA sequencing. We have Taq polymerase, which is derived from the thermophilic bacterium Thermus aquaticus. This Taq polymerase can resist temperature close to 95 degrees, and we have the DNA polymerase in E. coli which can resist up to 40 degrees, and also the thermally stable E. coli can resist up to 75 degrees. Then we have the RNA polymerase. RNA polymerase synthesize RNA. So in cloning, RNA polymerases such as T7 RNA polymerase are used to produce RNA molecules. So the T7 polymerase is for replicating the viral gene. So next up we have ligase. So ligase helps in the formation of phosphodiester bonds between the two molecules. The fragments may have like different ends when the nucleotides are cut off. The fragments can be blunt end or cohesive end. Here blunt end means we have this and cohesive end somewhat look like these. So here we have DNA ligase and T4 DNA ligase. The DNA ligase are isolated from E. coli, while the T4 DNA ligases are isolated from phage T4 bacteria. So these DNA ligases can form phosphodiester bonds between the cohesive fragments, and while the T4 DNA ligase form between both cohesive and blunt fragments. Since the DNA ligase are only forming the bonds between cohesive fragments, which are the same fragments, their ligation efficiency is better compared to the T4 DNA, where the T4 DNA is poorer because they are forming bonds between the cohesive and blunt fragments. So next up we have transferase. Transferases are used to add nucleotides, but only towards the 3' end. The type of transferase used for gene cloning is called terminal transferase. We have TDTA and TDTP, which are terminal deoxytransferase A and N. In the terminal deoxytransferase A, we have only the adenine group. So only the adenine group are added to the end of the 3' end. While in the TDTN, which is terminal deoxytransferase, we have adenine, guanine, cytosine, and thymine. So we can add all the mixture of nucleides to the end of the 3' end. So next up we have DNAs and RNAs. So we can see the difference between these two. The DNAs cut phosphodiester bonds in DNA while the RNAs do the same with the RNA. So the DNAs target more towards the double stranded DNA while the RNAs target more towards the single stranded RNA than the double stranded DNA. Next up, we have types of DNAs, that is DNase 1, 2, and MNase. Here the DNase 1 target more towards the 3' end. They cut phosphodiester bonds more towards the 3' end, and the DNase 2 do the same with the 5' end. Here the MNase does both. It cuts phosphodiester bond at 3' end and also at the 5' end. These MNAs are isolated from micrococcus. Next up, we have types of RNases, RNase A, C, and H. RNase A are little specific towards A and C, which is adenine and cytosine. Same with the RNase C, they are little specific with the C, which is the cytosine. And then we have RNase H, which separates DNA from RNA and also degrades them. So lastly, we have kinases and phosphatases. So kinases are enzymes which add phosphate group to the 5' end of the DNA molecules, while phosphatases remove the phosphate group at the 5' end of the DNA molecules. This is called the phosphorylation. So the polynucleotide kinase can add phosphate to any DNA molecule at the 5' end. So this is about the cloning enzymes used for cloning in genetic engineering. Thank you for watching this video.